actually, I'm really honored to have the opportunity to talk to you today. It is a real pleasure because I saw the birth of this community, of this society, and I'm particularly fond of it. And today I plan to talk about a subject, you see the title, that is extremely wide, that is across statistical physics and neuroscience. Necessarily, I cannot be exhaustive because the number of results is really huge, and I'll focus on a number of main milestones. Try to convey the general message that statistical physics can be very useful in understanding the properties of brain activity. In particular, the interest of the statistical physics community in the subject, I would say, was born in 2003 with the paper of Dietmar Plants. Here you see in the movie a slice of rat cortex with a microelectrode array where the colored dots are a measure of the intensity of the electrical activity. And so this is an experiment in a dish, is a slice of a cortex in a bath of glutamate, and people, so it's an activity of a neuronal system without any special stimulation, is an activity that takes place just because the system is alive. And for this reason, this is usually called spontaneous activity. In the human brain, spontaneous activity is about 50% of the overall activity, just to take your body alive. And in the literature for years, this spontaneous activity was thought to be a noisy signal, not so interesting in terms of the spatial and temporal structure. But then arrived this paper in 2003 where Dietmar Plants analyzed at the fine temporal scale the temporal signal, and he evidenced that this noisy stochastic signal was just the composition of bursts of continuously firing electrodes followed by temporal delays where no activity was taking place. He defined this sequence of firing a neuronal avalanche, and he measured the distribution of such avalanches. And astonishingly, he found that this activity was scale-free, had no characteristic size, and the distribution was a pretty robust power law with certain values of the exponents. First of all, pretty robust means that this behavior is found across species and across experiments. So it's found in vitro, in vivo, and ex vivo, and is found in vertebrate mammals, but also invertebrate systems. So initially he just cut a slice of rat cortex, but then he analyzed the brain of the rat in vivo and then the monkey, and here the bottom right figure is just the analysis of neuronal avalanches in MEG signals of healthy humans. So these are a signal for 13 women and almost as much men. So these are very strong statistical properties of this ongoing activity in the brain. Also dissociated neurons in a dish. This is a nice experiment performed in Genova, in IIT, exhibits the same statistical properties. The scaling of these distributions also is very robust if you adopt a unifying, careful experimental procedure to define the treatment of the data. And of course, there is a long story behind this statement. If people are interested, I can give you details. And the exponents of this distribution are consistently -1.5 and -2, that are exponents that characterize the mean field branching model universality class. And so the evidence, the this first evidence, then supported by additional experiments in different systems, for the first time suggested that in the rest state, when no task is performed, the brain can be considered as a system that acts close to a critical point because there is this absence of characteristic scale in the activity. So the activity shows complexity, but also the structure of the connected network shows complexity. And actually there are really two communities in neuroscience, one focusing more on the structure of the so-called connectome, on the connectivity where the activity propagates on, and the other on the properties of the activity itself. And the interesting result was that indeed this network is really complex. Here in the bottom right, you see the experiments of the late Ben Jacob in Israel, who let grow a network of connections among about 200 neurons of low caste in vitro. And after 7 days in vitro, these neurons establish connections that are the little lines, synaptic connections, so these are morphological connections that once analyzed, because they are mapped on a graph, they show small world properties. So the system is quite small, only 200 neurons, but still small worldness is a strong, robust properties evidenced in these systems. But morphological connections are not easy to detect for a system in vivo, because of course this is an invasive measurement. So often people analyze at macro scale, at the scale of the entire brain, rather the so-called functional connectivity. So the map of the brain is partitioned into small units called voxels, and the correlation coefficient of the activity for any couple of voxels is evaluated by the Pearson correlation coefficient. If the correlation coefficient is above a certain threshold, so there is this arbitrary threshold that must be chosen, you establish a connection. So the group of Eguiluz et al. Analyzed the functional network in healthy humans that were performing fMRI measurement doing a task that could be tapping a finger or else listening to music. And what they found out is that the degree distribution of the functional network has this nice property of being scale-free. So the connectivity degree is not homogeneous, but there is a strong presence of hubs in the system. But the other nice property is that regardless the value of the threshold chosen and regardless the condition, the different tasks, as you see, the scaling behavior of the degree distribution is universal. So there is this constant exponent -2 that is measured in all systems. So hubs are relevant, very relevant in brain networks, and successive papers by Bonifazzi et al. In SISA have evidence that hubs can be associated to inhibitory neurons. And this is also a very interesting result because, as you know, in the brain you have two basic kind of neurons, excitatory and inhibitory, depending on the kind of neurotransmitter they release. And in mammal brains you have about 20–30% fraction of inhibitory neurons. So already you have this first evidence that yes, inhibitory neurons are fewer, but they are highly connected. And then finally, I would like to mention a beautiful paper by Hernan Maxe in NYU about the fine structure of this functional network. The idea of Hernan Maxe was inspired in percolation. So Hernan also imposed a threshold to establish a connection between any couple of voxels, but instead of choosing a quenched value of this threshold, he started from very high values of the threshold, and then he was progressively lowering this threshold, adding in a hierarchical fashion first the very strong bonds and then the less strong bonds, and so on, until he was adding the weaker bonds. And what he found out by this construction is that human brain has a modular structure, and this was already well known, the existence of the functional areas in the brain, but from the network science point of view, he evidenced that these moduli, these functional areas, have a scale-free properties, are highly connected, are made of strong bonds, and have a scale-free connectivity degree, but they are isolated. Then, by the addition of weaker bonds, these isolated moduli become connected, and the overall structure becomes small world. So this is extremely interesting because stresses the role of the weaker connections, that are the one that connect moduli, are the one that are responsible for the information transmission from one module to another one, but at the same time, as we will see, they have a sort of contained role because they are weaker in strength. Useless to say that when all the experiments appeared in the literature, there was a lot of attention in trying to model and reproduce this activity. And already in neuroscience, there were a number of integrate and fire neuronal network model. By integrate and fire, I mean that each unit is a neuron that adds up all the stimulation coming from the presynaptic neuron that sends signal to this neuron, and then when the membrane voltage reaches a threshold value, a signal, an action potential is released, and this action potential is able to modify by a complex biomolecular mechanism the membrane potential in the postsynaptic neurons. A number of biological ingredients must be taken into care to reproduce experimental data, and I mean, for instance, the refractory time after firing a neuron remains quiescent, inactive, unable to respond for a time that is of the order of few milliseconds, but also the fact that connections, synapses are not static, but they are dynamical in time. So they undergo plasticity. There can be a temporary plasticity called short-term plasticity that is mainly related to the usage of neurotransmitter resources, but more relevant can be also the so-called long-term plasticity, the well-known Hebbian plasticity that is fundamental in the brain for memory learning. So all the learning routine and all the learning mechanism that take place in the brain use Hebbian plasticity. So here I show you results for the size distribution and the duration distribution of avalanches. As you see, they show a robust scaling behavior with a beautiful result that you expect in critical phenomenon at a critical point, that by changing the system size, the cutoff of the distribution moves accordingly. And in a class of models that have a tunable parameter, are not self-organized, you can actually tune the parameter so that you can actually move the system around the critical point, and you can also place your neuronal network below the critical point or above the critical point, so you can have also subcritical behavior or supercritical behavior that experimentally is observed in epileptic epileptic systems. To link to the information I gave you before, the modular structure of the network is really fundamental in the human brain, because you can actually see that implementing a neuronal network on a modular structure like this one, that has the same statistical properties measured by Hernan Max for the human brain, you actually see that on a modular structure, the cutoff size is no longer the size of the entire system, so the total number of neurons, but becomes the size of the larger module. And this is very interesting, because this implies that this modular structure, by this partition of weak and strong bonds within and outside the moduli, is able to contain constraint activity. So the information can pass, can be transferred from one module to the other. However, since the intramoduli, the connection between modules is weaker, the signal will arrive not with such strength to be able to propagate and invade the entire system. So you don't observe encompassing activity like in epileptic systems if the structure or the modular structure of the brain is organized as weak and strong bonds, organized as it is in the human brain. The observation of the scale-free behavior for neuronal avalanches and the suggestion that the brain could be considered as a system acting close to a critical point naturally arises another question, because criticality cannot be simply absence of a characteristic size. Criticality implies long-range temporal correlations. So a wide interest has also been given to the study of the spectrum of the signal. And the situation experimentally is quite rich, because in a variety of exponents, what is observed is that besides the characteristic frequency that are well known and corresponds to the brain modes, the power law of the spectrum in healthy patients as a value that is compatible with a 1/f noise. So the range of the beta exponent is in a narrow range about 1. The value varies weakly with the experimental condition and with the region that are examined by the experiments. However, what is striking is that if this power spectrum of epileptic patients has consistently an exponent beta equal to, so brown noise. So 1/f noise, long-range temporal correlations are typical of healthy brains, and this is in agreement with this evidence of critical behavior. However, epileptic systems that in our language can be named supercritical systems exhibit brown noise. And here I show you a first evidence of a result of a paradigm that is a pillar in neuroscience. And this pillar is the balance of excitation and inhibition in healthy systems. So if in numerical simulation you measure the power spectrum of the exponent, you find out that if your network is purely excitatory, the scaling exponent of the spectrum is -2, so you do have brown noise. However, by progressively considering inhibitory neurons, so trying to balance excitation, inhibition, and excitation, you see that the scaling exponent tends toward 1 when the percentage of inhibitory neurons becomes about 30%. That is the observed percentage of inhibitory neurons in mammal brains. So the fact that in our brain we have excitatory and inhibitory neurons is functional to realize, to implement the so-called balance of excitation and inhibition that is the key ingredient for healthy behavior. If you break this balance, pathologies comes in. And another nice observation that one can do at this point, and is of interest for statistical physicists, is that often, and in the literature, an analogy between the Barkhausen noise, so the avalanches found in Barkhausen noise, the so-called crackling noise, and neuronal avalanches has been proposed by a number of authors. And so the scaling derived by Jim Sekna in a number of papers for the shape of crackling noise avalanches and the neuronal avalanches have been regularly implemented to study data. However, the Sekna study of Barkhausen noise also say that this exponent, where this gamma exponent is written in terms of the scaling exponent of duration and size of avalanches, so these tau are 1.5 and 2 mentioned before, this exponent must be also the exponent of the power spectrum. So as you see, there is a strong disagreement between the value obtained for crackling noise and the spectrum of real brain signals in healthy humans. So the suggestion, this should raise the doubt about the validity of the analogy with crackling noise in neuronal systems, where really the mechanism of temporal organizations are more complex than just in Barkhausen noise in a Ising model. So the evidence of 1/f noise, or pink noise for neuronal signal, is already an indication of the presence of long-range correlations. However, you can further analyze this property, and in particular, we used a statistical approach that has been developed for years in the context of geophysics, has been applied to earthquakes in a number of studies. And in particular, the intertime distribution is the distribution of time delays between one event and the next one. So you don't care about the size of the event, you just care about the time you have to wait between one event and the next one. By this definition, is clear that these are statistical quantities that is very easy to analyze, and the advantage to analyze this question is that there are analytical results. So if the process is Poissonian, this distribution is an exponential, whereas if temporal clustering is present, this distribution shows a power law behavior. And here I showed you the data by Alvaro Corral on catalogs of California earthquakes, where you can actually set a threshold to the magnitude of events and evaluate for the different catalogs the distribution. This distribution consistently shows an initial power law followed by an exponential, so it's a gamma function, has a strong scaling behavior that is robust with respect to threshold, so is independent of the threshold. You just rescale the system by rescaling the average rate in the sequence, and you obtain the same functional form for this distribution. So we course, interested in verifying results for neuronal avalanches, also because results found for earthquakes turned out to be quite universal. They were found also in solar flares, in some acoustic emissions of rock fracture, in rainfalls, and so on. So our idea was to investigate the same property in slices of rat cortex. But as you see, this distribution now presents an astonishing new behavior that is non-monotonic, that is not found in other natural processes. And so here the behavior is typically, of course, with fluctuation from sample to sample. You have an initial power law decay followed by a minimum and then a bump with a maximum at about 1 second. Consider that the typical scale in neuronal system is the millisecond. And of course, a generic integrate and fire model is unable to reproduce this non-monotonic behavior. And in order to reproduce this result, one can actually point out again a different aspect of the properties of the property I was mentioning before, namely the balance of excitation and inhibition. So to reproduce this non-monotonic behavior, you must implement in your integrate and fire network of whatever kind, this is not relevant, the fact that the system undergoes an alternation of up and down states, so-called. So they are states of very high activity, where avalanches occur close in time, followed by states of almost quiescence, where no activity is going on in the system. In order to realize this macroscopic alternation of up and down state, you have to assume, and this is supported by experimental data, that the picture of a neuron that gets to threshold, fires, and go back to the rest potential is too simple, is not realistic. And indeed, a neuron can fire, but then set the value of the membrane potential not to the rest potential minus 70 millivolts, but remain close to threshold in order to be ready to fire back again after a short while. At the same time, in down states, the neuron fires, but then can go even below the rest potential, can even have a value of the membrane potential that is minus 90 millivolts, so minus 20 millivolts below the rest potential, and this occurs in the down state. This down state occurs when there is a general disfacilitation of the system due to the exhaustion of resources. If the system has fired a lot, a lot of neurotransmitter has been released, so neurons, a large number of neurons need to build up these resources again, and this takes time. During this while, the system is in quiescence. And so this non-monotonic structure reflects the up state, where avalanches are correlated in time and you have a power law, and then this bumpy regime with a characteristic size that is the typical duration of the down state. But what is interesting in this analysis is that numerically you can control up state and down state, tuning separately two parameters independently. But then what you find out is that no matter what is the value of each of the two parameters, what counts for the distribution that reflects the temporal organization of real system is the ratio between these two parameters. So this is a clear evidence that the system in a self-organized fraction, the system, the brain, in a self-organized fraction organizes the balance of excitation and inhibition, where balance now is a dynamic balance, is not a structural balance due to the fracture of excitatory or inhibitory neurons, but is just the alternation of activity and rest that in the healthy brain is done in the appropriate way, mediated, controlled by biomolecular mechanism like adenosine, external magnesium, and so on. So this is really a fascinating machine that controls the temporal organization of avalanches in time. And here you can see the comparison between the intertime distribution and the avalanche size distribution in numerical system and in real system, the slice of rat cortex, in healthy, the blue symbols, and pathological conditions. Pathological condition that occurs when you alter the balance shows an excess of large avalanches. You don't respect anymore, you don't fulfill anymore the scaling with the exponent 1.5, and also the intertime distribution is distorted by this pathological behavior. So this is an interesting understanding of the fine organization of avalanches in time. However, when this idea of criticality came into play in the field, a lot of criticism were raised by the traditional community in neuroscience that could not really understand how to reconcile the absence of a characteristic size of neuronal avalanches with the well-known existence of brain modes that are temporal oscillations in the brain signal. This intertime distribution is pretty powerful because it's also able to provide an explanation for brain oscillations, because if you now analyze the intertime distribution for the same signal of avalanches where you set a threshold, so you analyze only avalanches larger than a given threshold, that here is shown with the colored vertical bar, the corresponding intertime distribution changes dramatically. It's not like for earthquakes where you can rescale onto a universal curve, but you see that peaks develop, and peaks develop at the characteristic value of the intertime that correspond are the inverse of the characteristic frequency of the modes of brain oscillation. So the take-home message is that you do have avalanches without a characteristic time. These avalanches do not have a random or whatever temporal organization in time, but big avalanches occur according to the theta frequency, the low frequency brain modes, and between large avalanches there is a large number of small avalanches that occur at large frequency, gamma, and so you can actually by the organization of avalanches of different size in time, you can account for the so-called well-known brain modes. So let's now come to after this general introduction about spontaneous activity. As you see, we just focused on the activity of a neuronal system where there is no external stimulation. But now let's go into something that is of interest in neuroscience, because of course the function of the brain is to perform tasks. And so an open question was the relation between the spontaneous brain activity and the activity that the brain has once a stimulus is applied. The very first paper that attacked this issue was a paper by Ariely et al. That appeared in Science in '96. That was a really stimulating a breakthrough in the field, because what Ariely and co-workers show is that in experiments where a cat underwent a visual stimulation, that was always the same visual stimulation, so namely a moving grid, the response of this task, of this stimulation of the moving grid, was extremely variable. Even if the stimulus was always the same, the response showed a very high variability. So what they did, that they were able to do, they extract the average reproducible response due to the stimulation, and they show that the actual response of the brain of the cat to the stimulation was the superposition of two components. A component that is reproducible is due to the stimulus itself, so it's the first line. But then the second component is due to the state of spontaneous activity that the brain of the cat underwent when the stimulus was applied. So the large variability in the response was due to the large variability of the spontaneous activity of the cat before the stimulus was applied. And after this paper, a number of other experimental papers attacked this problem, and in all this variety of papers, there was, people showed the evidence of a strong correlation. This is a scatter plot of the average spontaneous activity, evoked activity, stimulated activity versus spontaneous activity. There is a strong correlation between those two quantities, with a correlation coefficient that is almost 1. So a neuroscientist then opened the door toward the conclusion that there is a strong relation between these two activities. However, no analytical, rigorous approach to this problem was proposed in the literature. And of course, coming from statistical physics, the idea we had with Alessandro Sarracino was to attack this issue within the framework of non-equilibrium statistical physics, and in particular the fluctuation-dissipation relation in non-equilibrium system, because the brain is a system, open system, and is a system out of equilibrium. And to attack in an analytical way the problem, not in a numerical way, of course, we could not use a model that was microscopic, so a model with individual neuron using a very large number of neurons. So in the literature in neuroscience, there is a model that is actually very suitable for this kind of analysis. And this is the stochastic Wilson Cowan model, that with respect to integrated fire models you saw before, is a model for a population of neurons. So the variable is the average activity of a population rather than the activity of the state of a single neuron. So here, I would like just to briefly summarize the procedure. So we take a system of an equal number of neurons, excitatory and inhibitory. A neuron can be active or quiescent and can change state according to the alpha. Alpha is the probability to become inactive once the neuron is active, and then there is the probability to become active, that is through an activation function whose argument is again the integration of all stimulation coming from the other neurons plus a small field that just hinders the collapse into a state with no activity, so can make the activity start again once goes to zero. And this activation function is an hyperbolic tangent. But then the interest is to write the number of active excitatory and inhibitory neurons in terms of a Gaussian approximation. So write this number of neurons as the sum of a deterministic component, E. So E is the average number of excitatory neurons that are active, and this component scales with n, and a fluctuating component that instead scales with the square root of n. This position is made for both population, for excitatory and inhibitory neurons. So the procedure, that is quite standard, starts from a master equation and then makes a Taylor expansion about the deterministic solution. So you consider only terms where the perturbation is small, the larger limit, and then what is instead a fundamental hypothesis is that the synaptic weights are symmetric. What do I mean by that? I mean that the strength of the connection does not depend on the nature of the post-synaptic neuron, but depends only on the nature of the pre-synaptic neuron. So an excitatory neuron will make the same connection, a connection with the same strength, with another excitatory or an inhibitory neuron. This hypothesis turns out to be fundamental for our results, as I will discuss at the end. So if you make this assumption, you end up with two systems of differential equation, one for the deterministic component and the other for the fluctuating component. However, studying this equation in terms of these variables is not very helpful because the comparison with experimental data is extremely difficult. You don't have access in a neuronal system separately to the activity of excitatory and inhibitory neurons. So it's also common to, in the literature, to rephrase this, to rewrite this equation in terms of new variables. And these new variables instead are very interesting because one corresponds to the global activity, sigma. The other corresponds, delta, corresponds to the unbalance in activity between the excitatory and inhibitory population. So you understand now the fil rouge with the previous discussion about balance of excitation and inhibition comes back again, this variable. And then in these two variables, you obtain in the above limitation approximation that I detailed before, what is called the linear noise approximation, again, two sets of differential equation. One for the deterministic component, this one, and by studying these two equations, you find the fixed point of the evolution of the deterministic component. And what is interesting at this point is that the fixed point for the variable delta is 0. So this system, by the sequence of the assumption we did, this model gives rise to a fixed point that realizes the balance of excitation and inhibition. So is a good candidate as a model to describe a system in a healthy state, close to a critical point. So this 0 fixed point is a direct consequence of the symmetry assumption we made for the strength of synaptic connection. What is then also, and will be the object of our study, is the other system of equations, the system of equation for the fluctuating part. And the coupling matrix for the psi variables has this interesting triangular shape. And this triangular shape, the presence of a 0 here, is also a direct consequence of the 0 fixed point for delta here and the assumption of the symmetry of the synaptic weights. So basically, what tells us this shape of this matrix? This 0 tells me that a perturbation in the fluctuating part of the sigma variable, psi sigma, does not affect the psi delta. Conversely, a perturbation in the psi delta will affect the psi sigma. So a fluctuation in the global activity does not affect globally the balance, but a fluctuation in the balance will affect the global activity. And this non-zero term that is called the feed-forward term, we just tells us how the fluctuation in delta will affect sigma. And so this system of linear Langevin equation at this point can be integrated. So now we have the solution for the two psi, for the vector psi, where here we call M as coupling matrix, this triangular matrix here. The two diagonal term conversely will end up to be two inverse characteristic time, as we will see in the next slide. And the eta is a noise that is delta correlated. So this can be integrated, and this is the solution. But then we are ready to perform the analysis of correlation, and we can calculate the correlation function for the variable delta. That is as 4 terms, and so the general expression is this exponential matrix with a coupling triangular matrix here, where sigma is the covariance matrix, that is the 0-time correlation coefficient. If we want to compare the results of our analysis to experimental data, of course, of the 4 correlation functions, because you have sigma sigma, sigma delta, and delta sigma and delta delta, we have to focus our attention on the sigma sigma. So here I write you only the expression of the correlation function in global activity that we want to compare with experimental data. And you see that this correlation function has a nice double exponential decay, and we then looked for experimental data to verify the analytical prediction, and we were able to obtain data for MEG on healthy patients, and these were obtained in Ben-Gurion University in Israel by the group of Orange Shriki, and this is the correlation function of experimental data. You see different symbols, and for a reason that will be clear in the next slide, we analyze both the global activity, so the activity averaged over all sensors in the system, but then we also analyze separately the occipital area and the temporal area, for a reason that I said will be clear shortly. All these data sets are consistent, show a very good fit with a double exponential decay, where you have a first characteristic time that is quite short, of the order of 10 milliseconds, and the longer characteristic time for the second exponential. This is the average over 7 subjects, and this small peak here is the reminiscence of the brain modes, is coherent with the theta peak, so is something you don't find in the analytical solution, but you find in experimental data. But then also for data for a single subject, there are kind of noisier in the occipital area and in the temporal area. You consistently fit the data with the double exponential. So even the simple linear noise approximation of this Langevin equation is able to provide an analytical expression that fits data from the human brain. But then this is the major aim of our study, the formulation of a fluctuation-dissipation relation for the brain. And following the procedure that has been developed in the recent years by many groups, from the group of Angelo Vulpiano in Rome, Letizia Cugliandolo, the group in Salerno, and so on, we are able to derive the response function, and the perturbation is instantaneous in our analytical derivation at times t equals 0 in the j variable, in the xij variables, and we observe the response in the xii variable. And analytically, the form of the response function is this one, so can be directly related to the correlation function in spontaneous activity through this covariance matrix sigma. And of course, as in the previous case, we focus on the global signal, and now it's clear why we focus our attention on occipital and temporal area, because the same subject whose spontaneous activity we analyzed in the previous slide underwent visual stimulation. So they underwent a sequence of phases, exposure of phases for a duration, the exposure of a second, separated by a delay of some seconds. With respect to the analytical solution, of course, the simulation is not instantaneous, and of course, we have no way to decide to say if the exposure of a phase can be considered a small perturbation. That is instead the hypothesis in the analytical solution. But then, if we calculate analytically, if we focus on the R sigma sigma, the general expression that has two terms, an autocorrelation and a cross-correlation, simplifies into a simple exponential decay. Why? Because of the complex form of the M matrix, the triangular form of the M matrix, the second exponential cancel out, and so the response function is just a simple exponential decay. So we analyze experimental data. This is the average, and again, as you see, the signals are very similar either for the global activity or the occipital area, where the visual functional area is, or even the temporal area, where there is the area that is in charge of phase recognition in the brain, the so-called phase fusiform area. So either the localized signal or the overall signal show consistently the same functional behavior that is a simple exponential. Or the prediction analytically is that this characteristic time should be the same time of the first temporal decay, so should be of the order of 10 milliseconds. Instead, the experimental fit provides a number that is larger than the one theoretically expected. However, there are a number of approximations that we were obliged to make to analyze experimental data that you can easily imagine in this bottom left figure. These are the 6 different subjects, and you see that the stimulation is applied at t equals 0. Every subject has a different latency and is a different maximum amplitude in the response. So we had to make the simplification, analyzing only the signal from the maximum value and the exponential decay. In the analytical solution, no latency can be considered. So the take-home message of this study is that the functional behavior, first of all, appears to be scale invariant. Either you analyze the global signal or you restrain to a particular area or even to a single sensor. These are data for single sensors. As you see, they are extremely noisy. However, they are consistent with an exponential decay. There is therefore an indication for scale invariance. The qualitative agreement is fulfilled. However, to get a good quantitative agreement, we have to try to perform, which we will try to do next, more controlled experiments where the stimulation is really short in time and weak in amplitude. So we are hopeful in trying to clarify this point, or else the alternative is to make a more general derivation without making use of the symmetry of the synaptic strength. In this case, the coupling matrix will not be triangular any longer. The cross correlation term in the response will not disappear, and so a richer comparison with experimental data will be performed. But in any case, the take-home message is that there is an evidence that information in the response of a brain to a stimulation, in the response that the brain gives to a stimulation, is contained in the state of the brain before the stimulation is applied. So I think I have abused on my time. As a last thing, I would like to thank all the people I've had the pleasure to work with, experimentalists, theoreticians, and a number of students in blue. Thank you very much.
Okay, thank you, Lucilla, for the very interesting talk. And now we have time for questions, if there are any. So I see that Stefano Ruffo has a question.
Okay.
Lucilla, in which cases do you get a single exponential in the decay, and in which others you get a double exponential? Because there are different experimental conditions, or model conditions?
You mean theoretically or experimentally?
I mean both. So I would like to understand if you could, in fact, give a unified picture where you have a transition from one to the other.
In correlation functions, there is a strong evidence for a double exponential decay. Remember that this is a system, this is a Taylor's function close to the fixed point, so close to the critical balanced fixed point, so LT systems. If the system becomes supercritical, so you break the balance, you can simulate this in neuronal networks, oscillations appear on the second exponential. But if the system is healthy, exponentially and numerically and analytically, you have a double exponential decay. That is this one. The response function is analytically is the sum of these two terms, but once you substitute this autocorrelation and cross correlation, the second exponential, the one with the exponent e to t to minus tau, cancels out.
I see. Okay.
And so you end up only with a single exponential. That, in my understanding, is nice. That is the exponential with a short characteristic time. See, this is τ1. τ1 is the shorter of the two. Why is nice that is like this? Because you know that human eye can appreciate about 10 images per second. If you had a decay over 600 milliseconds, you could not discriminate so many images in a second. So because you had the overlap between the response to different stimulations. So frankly, I understand and I like very much that this decay is of a short characteristic times, because this accounts for the performance of the human eye that is very high. On the other hand, if I simply take the data, the data for the single subjects are like this. I know the time where the face is shown to the patient, and this is the time t equals 0. So as you see, the peak in the response is a delay in time of about 200 milliseconds, no, maybe 120 milliseconds. So there is a latency, also because there is a memory. After a while that this patient sees faces, in a way is already expecting to see the face, and this also changes the response. So in principle, this experiment should be done with a very short instantaneous stimulation and a long delay in order to lose memory, and so to make another stimulation and so on, no? Because the same subject shows different response after a while. But then, since these were the only data we had, we forgot that the stimulation is applied at t equals 0. We just took the maximum, and we analyzed just the decay from the maximum. And these are the data that are here. And these are the experimental data.
Thank you.
Okay. Also, Roberto has a question.
Lucilla, I have a curiosity. You stressed very much at the beginning of a talk the very importance of the inhibitory component with respect to the excitatory, in the sense that only balance between these components can allow a healthy behavior of the brain.
Yes.
I never understood correctly, probably because I'm a bit stupid, but in some sense, this percentage, which is fixed for human brain and other brains that work usually under proper conditions of inhibitory neurons, is on one side a sort of built-in physiological property of brain, but on the other hand, also a dynamical, this is just a guess I do, a dynamical training of the neurons should decide, which is the history. For instance, it's very well known that at the beginning, if you have an animal, he has not already produced these specification of neurons during his growth. Only at the end of a maturation process, this comes out. So how much is this really relevant? This mechanism of specialization is really relevant to understand the behavior of a healthy brain.
This is a huge question. Of course, evolution from the critical period of the brain, I mean the initial days after you are born, to the adult age also implies differentiation in this direction. In the adult, for a long time, people thought that this percentage of excitatory and inhibitory neurons was fixed, and about 30% in mammals' brains. However, in the last 10 years, in the last 10 years or so, has become evident, and now is very well accepted, that in several conditions, also in the adult, you can have neurogenesis. So new neurons can be by utilization of stem cells, can be born in different areas of the brain, not only the hippocampus. So to to answer your question is very difficult. However, what is impressing in the brain is the ability of self-organization. So also the alternation of up and down states, that is a dynamical tuning of excitation and inhibition that is done on the basis of the quenched fraction of neurons, because it acts on very different time scales. Also, this is a measure of how important is this balance for the behavior of the healthy brain. I think this is very much a work in progress, because
do you believe that this balance is really relevant in relation of keeping the system close to criticality?
Yes, this is my understanding.
Ingredient. This is a crucial ingredient.
Fundamental, yes.
Okay, thank you very much.
Okay, I see that Tommaso Gili has a question.
Thank you. Very interesting talks. I was wondering, maybe you told it and I missed it. Why do you think? Would you think in the RT plot of the fluctuation-dissipation relation, there are so many outliers coming from the occipital area, respectively the fit? The plot.
You mean the correlation function or the response function?
No, the response function.
You are talking of the single sensor, so you talk here in the response function is certainly noisier than the correlation function. Also because the correlation function is a very long temporal signal. The patient is at rest. Okay. You have a very long signal. You break it in windows of 1 second long. So in this analysis, there is a very strong statistical average. Yeah. In this analysis, instead, we are constrained by the number of exposures, that is about 500 phase exposures for each subject, and there is a strong variability from subject to subject and from exposure to exposure. And so this is certainly, that's why I think controlled experiments where one really tries to adopt a protocol that reduces the experimental variability would be important, like, exposing phases every, 5 minutes, not every 5 seconds. Because already there is a sort of training of the system after seeing a large number of phases. The response is not independent from the previous one. So you already see from these six signals what kind of variability we have. If this is the fluctuation you were, the fluctuation you were referring to.
Yeah, when you say that subjects were at rest, you mean with eyes closed or with eyes open?
With eyes closed, but without sleeping.
Okay, thank you very much.
Mm-hmm.
Okay, any other questions? Okay, so since there are no more questions, we can thank again the speaker for the very interesting talk. Thank you.
Thank you.
Thank you.
Ciao.
Ciao a tutti, io sono al buio.
Ciao.
Saluto.
Ciao.
Ciao.
Ciao, ciao.
Grazie Lucilla.
Siamo a
Ciao.
presto. Caro, grazie a tutti. Ciao.
Ciao.